Welcome to Ask the Expert. This is a brief, informative, and lively discussion with experts in type 1 diabetes and related interdisciplinary research. We're recording this event. We're going to post it on our Sugar Science YouTube channel shortly after the presentation. And if you have questions for our guests, two guests today, please feel free to enter them in the chat or raise your hand at the end of the presentation. So today we have two guests. We're very lucky to have Dr. Jennifer Wang. She's a renowned physician scientist in the field of type 1 diabetes and viral immunology at UMass Chan Medical School and UMass Memorial Health in Worcester, Mass. She's committed to understanding innate immune response to viral infection and how innate immunity contributes to the etiology and pathogenesis of type 1 diabetes. And she's made a huge contribution to the field of infectious diseases and immunology. She has more than 60 peer-reviewed publications in many high-impact journals, Cell, New England Journal of Medicine, Nature Communications. So welcome, Dr. Wang. And we also have Dr. Decio Eizirik, MD, PhD. He's presently a full professor and director for the Laboratory for Experimental Medicine and Center for Diabetes at University Libre de Bruxelles in Brussels, Belgium. He's originally from Brazil. He's had a long and very illustrative career in the field of type 1 diabetes. He's spanned positions at UBC, Uppsala University, and recently at Indiana University. He has a brand new couple of papers that are really interesting, and we can't wait to hear about them today. I'm particularly interested to hear a little bit, a taste from the paper in Nature Communications. An integrated multiomics approach identifies the landscape of interferon alpha mediated responses of human pancreatic beta cells. I hope he's going to just touch on that. And today, really the big sort of the discussion is really going to center around the role of viruses in the etiology of autoimmune diseases, specifically type 1 diabetes, and the ultimate target, beta cells, pancreatic beta cells. So welcome, Dr. Eizirik and Dr. Wang. I wondered if you could just tell us a little bit about yourselves. Just first, Dr. Wang, if you'd like to just sort of go first and talk a little bit about sort of your background.
Sure.
Thank
you very much.
Sketch of your career path.
Yeah. So, I started out as a clinician in infectious diseases, really driven by HIV care, then progressed to research-based career with a focus on innate immunity and various pathogens, eventually settling in on viruses, including Coxsackie virus, influenza, and others. And so my interests in autoimmunity and type 1 diabetes were, initiated about 10 years ago. And really with the GWAS findings of RNA sensors being associated with T1D, this prompted my interest. And it's such a rich field with immunology, and I find it very exciting. And then this past year and a half, I've been focused largely on COVID, both the patient treatment and prevention, and research in the laboratory. And interesting to reflect on the analogies between autoimmunity and COVID.
Yeah, for sure. There's going to be some interesting data coming out of this unwanted experiment with COVID. Dr. Eizirik, can you give us a thumbnail sketch of your sort of background and intersection with type 1 diabetes?
Thank you very much. Well, my background is a little bit similar to Jennifer. I also started my career as a clinician. I was trained as in internal medicine and endocrinology, and I then grew with particular interested in diabetes treatment. And interesting or sad enough, my first research area were long-term complications of diabetes, particularly nephropathy and autonomic neuropathy, which are devastating complications. So based on this experience, I got the feeling that perhaps we should try to intervene at an earlier stage of the disease before this type of complication appear. And then I engaged in a very long career, try to understand the pathogenesis of type 1 diabetes. And I took, when I started up to 5-6 years ago, most focus in the field was in the immune system. But I always looked at from the other side. I looked instead of looking at the assaulting, let's say, the immune system, I looked at the victim, the beta cells. And as the years went by, it became clear that there was a very complex relation between the beta cells and the immune system. And most probably there is a dialogue between these two systems that, given a particular genetic background, may lead to a misunderstanding and eventually evolution from innate immune response to adaptive immune response and autoimmunity. And in this process, also in the last 3, 4, 5 years, I grew more and more interested in type 1 interferon. So I really look forward to listen to Jennifer and to start this discussion today.
Yeah, I mean, this is going to be a great discussion. I'm looking forward to hearing it as well. Let's sort of dive in. I know you both have some slide sets, and let's sort of start with Dr. Wang starting to walk us through her beautiful slide at the beginning. I love this immunofluorescence visual. It's gorgeous. So yeah, take it away.
Thank you. So I have about 10 slides, and as I had originally put this together, I realized that, I'm looking at more modeling and in animal studies, and I really wanted to show how this complements human samples and our current understanding. And so I think the two talks are very complementary. So this first imaging I put up here, this is a rat pancreas in the pre-diabetic stages, and it's RNA scope, RNA in situ hybridization. And you can see the green here is insulin, and in this case, the yellow is CXCL10 and red is glucagon. And we can look at entire sections by scanning and zooming in and probing numerous different targets. So the approaches that we use in our models is this combination of RNA in situ hybridization and single cell RNA-seq. And the fundamental questions revolve around what are the innate immune signals that can be in the early phases of diabetes, type 1 diabetes, that lead to recruitment of the adaptive side and the onset of disease. And I'll describe the model. And again, the importance is how well this may correlate with human type 1 diabetes. All right. So many of you have seen this list of genes with risk genes associated with T1D, certainly HLA, but also in the last 10, 15 years, a number of other genes have been identified largely through GWAS, and these include innate immune sensors such as IFIH1, which detects double-stranded RNA, also other genes important in interferon signaling and regulation. And this, I put up a couple of different examples of how this is similarly seen in human disease, these type 1 interferon signatures. So this is a growing area, and my interest is in using a rat model of T1D to further probe the innate immune responses and whether these could be targeted towards type 1 diabetes prevention. So there's a model which has been highly used at my institution, the Lu1W1R rat, as well as BBDR rat. They're very similar in their findings. They are both associated with MHC class 2. And these are inbred rats. There is a juvenile onset, which parallels somewhat with human type 1 diabetes. There is no sex bias with preference over males or females, unlike other models, such as in the mouse. And the insulitis that's seen on pathology is similar to that in human pathology. So these are all advantages. We can, because these are animals that are inbred, we can sample multiple animals over time and collect different specimens, which, as you know, is a challenge in human studies. So my interest, again, was also prompted by that this is a virus-induced diabetes model, and intrigued me. How is this happening? And the particular virus is a rat parvovirus. And interestingly, it doesn't have to, this model can develop even in the absence of virus. One can add a double-stranded RNA mimetic to induce diabetes. So there's something about the innate signaling rather than the infection per se. So we can look at this over time, and my focus has been to look at these early times in the pre-disease onset to really look at the development. And so we then used a CRISPR-based targeting to examine the question, what is the role of the IFNAR receptor in contributing to autoimmune diabetes in this model. So we made the knockout, and we found that in this particular, this was published 4 years ago, if we trigger the type 1 diabetes with multiple injections of poly-IC, then in a wild type animal, LU1W1R rat, there's nearly 100% diabetes. And then in the knockout, where there's no functional IFNAR, then there's a significant degree of protection, although it's not 100%. There is still some breakthrough. And in follow-up, we had looked at some immunohistology in that first paper, but to really understand even the earlier immune events, we focused on day 5 spleen as a source of immune cells, and also because this is a site, we use the KRB infection model for these studies I'm about to show, and the virus actually goes to the spleen at high concentrations and replicates, and so presumably there's a lot of viral intermediates of replication triggering innate immunity. And so there's a balance hypothesis which has been proposed, where there needs to be an appropriate balance between T effectors and T regulatory cells. And it's been described that the virus sets off this balance and leads to onset of diabetes. And in the past, there has been a loss of Tregs and increase in CD8 cytotoxic T cells. When we examined our model, and we did a combination of flow cytometry, bulk RNA sequencing, and single cell RNA sequencing, we found this to be the case as well. We did see an increase in NK cells in this model. And then when we looked at the IFNAR1 deficient animals and challenged them with the virus, there was a restoration of this balance and protection against diabetes. And appropriately, we see a decrease in interferon stimulated genes. We also see an increase in macrophage MHC2 expression. So this is an example of what we can learn from the rat model. And then more recently, we have been focusing on examining the pancreas in the event of the pre-diabetic time course. So here's, we can isolate islets from the rat pancreas and dissociate and perform, in this case, 10x sequencing, and we see the appropriate distribution of cells. And what we've done is taken rat samples over time course to see the development of signatures of any immunity in the cell populations. And just as a highlight, we can focus on the beta cells. Here, they're all highly expressing insulin transcripts. And what we see, the bottom line is that there's a strong innate immune signature in a subpopulation of cells. And this signature, we're in the process of validating this through various methods, but heavily, we see this CXCL10, as I showed on the first pathology slide. So our current understanding is how, why is this only a certain number of beta cells, the distribution over islets across the whole pancreas, and then how does it relate to immune cell recruitment? And we can look at this by sequencing, where we see this type 1 interferon-based signature increasing over time in the pre-insulitis period. And then we see a recruitment or an increase in the total number of T cells from our sequencing data, and also an increase in macrophages. And using immunostaining and RNA in situ hybridization, we can see that these quote unquote hot islets, hot beta cells, are indeed recruiting the immune cells. So, just in summary, this is our inducible rat model, which we can introduce knockouts to try to understand important host factors in the development of autoimmune diabetes, and look at this time course of development and potentially identify pathways that we can target through therapeutics. And this, I'll show you the next slide. What kind of drugs do we currently have available that could be used for modulating the host immune response in somebody, say, who is at high risk? And Monica brought up, that anifrolumab has been recently FDA approved for treatment of SLE. And there are other, this is specifically targeting human IFNAR. And if a candidate has moderate to severe lupus, they received over a year monthly administration in this particular study, that there was an overall reduction in disease severity. There were anticipated side effects, although none of them were severe, and there was a significant increase in the number of viral infections as expected, mostly upper respiratory, bronchitis, nasopharyngitis, and also reactivation of herpes simplex virus, and that was significant. So this is from Decios' recent publication in Diabetes, and I thought this was a good opportunity to highlight the different druggable targets. So IFNAR is here, and then certainly JAK1 inhibitors such as baricitinib. These are things that we use in COVID and in the later stages to try to blunt the overall immune response and the hyperinflammatory syndrome. And this, could be a potential target. And then I'm going to hand it over to him because, there are multiple downstream pathways, and he'll talk about human data in these pathways.
That's fantastic. I mean, it really just goes to show that the rat is still a very viable and useful model system for exploration. Thank you so much.
Thank you.
Okay, so now we're diving into the human beta cells. And this recent, I think he's going to talk a little bit, Dr. Eizirik is going to talk a little bit about this paper that you just referenced, and I'm looking forward to hearing that too.
Thank you very much, and thank you very much, Jennifer. I think you situated very well the problem, so I don't need a very detailed introduction. So, as Jennifer said, one of the things that made me also think that type 1 interferons are important in the pathogenesis of type 1 is when you look at the candidate genes. So you have nearly 50% of the candidate genes for type 1 diabetes are expressed in human beta cells. And when you start to look at the function of these candidate genes, you find that many of them are involved in the innate immune response, either in the direct recognition of a viral infection, for instance, MDA5, or in the signal transduction of type 1 interferons. So as Jennifer said, you have TYK2, which is a kinase, and for instance, patients or individuals who have a polymorphism that decrease TYK2 expression, they are partially protected. Then you have a phosphatase like PTP and N2 that will dephosphorylate STAT1, STAT2, and also inhibit J and K, and several other candidate genes that act on this pathway. And a common motif in these candidate genes is that many of them have the role of downregulating an innate immune response, which starts to get the feeling that when the beta cell over-respond, for instance, to a viral infection, this is bad news, and we can discuss this at the end. So we grow curious about the impact of interferon alpha on human beta cells, and we decided to do a time course multi-omics analysis of both a human cell line, end of beta H1, and whole human islets of Langerhans. So we did in parallel RNA sequencing, ATAC-seq, which allow you to see which areas of the chromatin are opening. And at 8 and 24 hours, we also added proteomics. And then after this data became available, we did a detailed data integration. One of the things that is important when you are using this type of in vitro model is check whether it is indeed of relevance to the human disease. And the way we approached this was to look at RNASeq, which was actually done by colleagues from Jennifer, David Harlan, and colleagues, where they were able to obtain FACS-purified beta cells from 4 patients affected by type 1 diabetes and 12 controls. So we compared the gene expression induced by interferon alpha treatment as compared to the gene expression induced in type 1 diabetic patients, compared against control. And in red, you have the intensity of the significance, and here you have the upregulated genes. So you can see that there is a highly significant concordance in the upregulated genes in the human islets treated in vitro with interferon alpha and in the beta cells exposed, let's say, in vivo to the type 1 diabetes environment. There is little similarity in the downregulated genes, probably because the interferon alpha treatment was brief, just 18 hours, which is not enough to induce a severe stress. And this is probably specific because when we compare the gene expression of the human islets treated with interferon alpha with the gene expression of islets from patients affected by type 2, there is very little correlation. So this seems to be specific for the type 1 situation. So when we started to go a little bit deep into the data, the first thing that struck that was really interesting was when we look at the attack sec, how fast the chromatin opened. And indeed, there is much more opening of chromatin sites around 4,000 genes at 2 hours than at 24 hours, where there were around 1,200 genes. And this makes sense because type 1 interferon is an early response protein to a viral infection. So the cell really needs to react fast, and it makes sense that there is this massive early change in chromatin. The other thing that was interesting, and this we observed both with interferon alpha and in a collaborative study with Lorenzo Pasquale, was that when we look at, let's say, at the transcription factors that were binding to these opening sites, we were surprised to see that there were co-binding of transcription factors. For instance, you had a binding of a transcription factor like neuro-D and so on. Let's say neuro-D, PDX1, FOX, they are all genes involved in beta cell phenotype maintenance. And what we found is that there was co-binding of these beta cell phenotype related transcription factors and the new, let's say, inflammation related transcription factors like STAT1, STAT2, IRF1, which were induced either by interferon alpha or by interferon gamma, suggesting that the fact that the chromatin in some cases already open and bound to some transcription factors like PDX1 make it easier for transcription factors like STAT1 to also bind. The other thing that was interesting is that in general, when we think about the effects of pro-inflammatory cytokines on human beta cells and rat beta cells, we always think on the negative impact that the cytokines may have. But we found that they also, for instance, interferon alpha, induce several proteins that really inhibit the immune system. One of them is PD-L1, which is highly upregulated, and we indeed confirmed that islets from patients affected by type 1 diabetes who still contain insulin expressing beta cells have a major increase of PD-L1 and also of HLA-E, which is an atypical HLA type which inhibits the immune system. So the impression that it gives is that the beta cells are trying, at least in the early stages of the disease, to defend themselves from the immune system. And this may be clinically relevant because, for instance, checkpoint inhibitors that are used to treat cancer that block PD-L1, in 1 to 2% of the cases, they may trigger type 1 diabetes mellitus. And it's puzzling because 9 to 10% of these patients may express or may suffer also from other autoimmune endocrine diseases, including hypophysitis, which is extremely rare. So one interesting question here is why particularly endocrine glands are susceptible when you block PD-L1. Well, we then did the analysis of the pathways involved, and you find, for instance, regulatory mechanisms like PD-L1. Of course, you find interferon signaling, ER stress, and so on. And the next step, we mined these data sets using available data sets at the Broad Institute to find drugs that could antagonize the signatures that we were observing. And one of these drugs were inhibitors of JAK1 and TYK2. And as Jennifer mentioned, both JAK1 and TYK2, they are kinases that signal downstream of the type 1 interferon receptor. And there are drugs being developed for other autoimmune diseases that target JAK1 and TYK2, but we were particularly interested in TYK2 because, as I mentioned before, polymorphism TYK2 that decrease its activity have a protective effect against disease. And there are accumulating data to suggest that drug development programs supported by human genetic evidence are more likely to be successful. So we started to test some of the TYK2 inhibitors that are used, for instance, in psoriasis, and we saw that they inhibited interferon alpha-induced HLA class 1. They inhibited this chemokine that Jennifer mentioned, CXL10, and here is a complete inhibition. They also inhibit ER stress. And when we did histology for MHC class 1, you can see under basal condition in a human islet, you don't see MHC class 1, or very little. When you expose this islet to interferon alpha, there is a clear-cut induction, which is inhibited by the two TYK2 inhibitors that we test. And we now are testing this together with Carmela Evans-Molina in mouse models of type 1, and we are getting some interesting data. So it may be the TYK2 inhibitors, which are already being used, for instance, in psoriasis, may be an interesting drug to consider as an early treatment for type 1. So the conclusions, or preliminary conclusions at that stage, are that human islets obtained from individuals affected by type 1 diabetes, but not type 2 diabetes, present a gene signature that is similar to that induced by in vitro exposure to interferon alpha. Interferon alpha promotes early changes in chromatin accessibility that are probably necessary for a rapid response during a viral infection. And the exposure of human beta cells to interferon alpha or to interleukin-1 beta activates several cis-regulatory elements, many of them presenting enrichment for islet-specific transcription factor. So these primed regulatory elements may facilitate cell type specific responses to pro-inflammatory signals, potentially explaining tissue specific susceptibility in autoimmune diseases. And the last point is that testing perturbagens. Perturbagens are drugs that antagonize the signatures that we have detected, may suggest drugs that can be used or repurposed for the treatment of different autoimmune diseases. This is what I would like to present, and I will now be happy to discuss together with Jennifer. Can I start asking a question to Jennifer?
Yes, absolutely. And anyone is welcome. Any one of our participants are welcome to also add a question to the chat, or you can unmute yourself and ask a question. Feel free.
Jennifer, I was really puzzled by your single-cell RNAseq, where you showed this heterogeneity in CXL10 expression.
Mhm.
Well, this is not spatial, but would it be possible for you to see if these cells that express more or less CXL10 have, for instance, higher expression of PDX1 or MAFA, so that there is any correlation with the beta cell function?
Right. We could certainly do that, and we're getting more experience adding these additional probes to see, where there are alterations, whether increases or decreases in specific transcripts, and specifically where they are in the beta cells and islets. So yeah, I'd be interested to know, what you think the most important targets are, the questions.
Yeah, we are doing also single-cell RNA-seq, but we are using iPS-derived human beta cells, or pre-beta cells. I wouldn't call them beta cells. I will take a look if there is this heterogeneity in CXL10. We are trying to see, let's say, if the different signatures are related to function of the beta cells, but we have no data as yet. Many years ago, we did a study together with Danny Pipeles, but this was in rat beta cells. And what we observed is that the most active beta cells in the rat were the most sensitive to interleukin-1 beta. So these were the cells that died first. But I don't know if this is true in human beta cells. And I think with single cell, this can be perhaps clarified.
I have a question for you. And as far as, what do you see as, things that need to be resolved or barriers to, initiate trials for, say, baricitinib or anti-IFNAR in prevention of type 1 diabetes in people?
This is a great question.
I think, in my view, the main issue is to convince big pharma to give these drugs and to sponsor this type of trial. We have been pushing for TYK2 trials, and one of the issues that we see is that since these drugs are already being tested in other autoimmune disease, there is a resistance to test them in the early stages of type 1. So I think the main issue here is to convince our colleagues in pharma that is the moment to start testing these drugs in the early stages of type 1.
How might T1D exchange help in this quest, or could they?
Well, we have in Europe Inodia, which is doing something similar to the trial net, and Inodia is already doing several clinical trials. So we have, it's directed by Chantal Mathieu, we have the whole infrastructure. What we need is the drug and the support. So I hope this will eventually come. But I think there is already a trial in Melbourne of JAK inhibition, JAK1/2, and they are recruiting. Tom Kay is leading it. So it will be very interesting to see what happens.
My experience, over, certainly in the past year and a half is that, I'm involved with about a dozen clinical trials, preventive and therapeutic for COVID. And certainly there's a lot of investigation on anti-inflammatories because that's where we see, the severe disease and high mortality. And I can tell you, multiple companies, pharma is highly interested because, and there's just, say we have an ongoing trial sponsored by the NIH for prevention of more severe COVID in hospitalized patients, and it's comparing different anti-inflammatory agents. So, approaching the NIH, I've become very familiar with many of these drugs, and after this, wave of COVID and the pandemic abates, that may be a good time to, approach the companies and say, look, you did a great service in studying this. How about we move to other diseases?
Yeah, and I guess the COVID came with a sense of urgency, right? And so somehow we have to, I think, as a community, extend that sense of urgency around type 1 diabetes as well, because it's a lifelong, burden to carry. And with diagnoses starting earlier, 2 years old, 3 years old, that's your entire life that you're carrying this 365 days a year, 24/7 disease. There a lot of changes have made it much more manageable, but it still has to be managed. And so it would be a tremendous win to use some of these resources and partnerships that came out of the COVID crisis to sort of attack it there. I wanted to bring up something, I heard a talk by David Harlan, who's a phenomenal scientist, and we talked about this a little bit earlier. His comment was, what kind of immune system, doesn't kill all of its targets, and why is there such a patchiness in the apoptosis events or the killing of the beta cells? Why is it such a patchy disease? And I mean, I guess I would say, when we think about what you guys are talking about, how does that fit with your model? Why do only, some beta cells change their chromatin accessibility? I mean, why is it, how does it fit your model, I guess?
Yeah, we're certainly asking that, why is the signature, to start, only in a very small fraction? What's driving the increase? And, how does it associate with beta cell function? And, what halts it eventually, or it prevents every single cell? And, these kind of on-off questions are critical and, are key to preventing further disease and, any disease rather.
Yeah, well, I think there is a short answer and a long answer. The short answer is we just don't know. The long answer, there are several hypotheses. One of them, as Jennifer says, function. The other is that, for instance, the expression of PD-L1 and HLA-E, which may be protective, is very heterogeneous. So it may be that beta cells that express higher levels of PD-L1 are better protected and survive longer. The other possibility is that some of these beta cells under fire, they differentiate and express less antigens, so they become less attractive to the immune system.
Would you say that they become almost senescent, sort of to talk about, a little bit about Anil Bhushan's hypotheses?
I must say that this senescence story, we have looked at in the RNASeq of type 1 diabetic patients and patients with type 2. We see clear signals of senescence in type 2, but much less in type 1 in the actual patient material. So in type 1, I don't know how solid is the role of senescence. What I was thinking more is a loss of function once they are under stress. I think senescence needs additional studies.
Yeah. There was also some talk, we had the State of the Science 3 weeks ago, our State of the Science number 7, Kevin Harold was on. He was talking about, he inferred that, we have to look more closely at the actual innervation of the islets, and maybe that even plays a role in their response to inflammation and viral impact.
I think there is one question by Connie, and for some reason she sent to me only.
Okay.
I don't know if you can see it. Connie Giesen.
What does she have?
She's asking JAK1/2 inhibition is being explored in type 1 diabetes prevention. So what would be the additional benefit of TYK2 inhibition? I don't think there would be an additional benefit. I think you cannot block too much these pathways, because then you make the cells and the whole patient too sensitive to a viral infection. So for instance, when we are trying to inhibit TYK2, we try to inhibit around 50%. And we have shown that at this level, they remain not resistant, but they don't become more sensitive to a Coxsackie infection. So I don't think it would be wise to block both JAK1 and TYK2. Probably you need to go for one or for the other. One advantage of JAK1 is that it may have more impact on the immune system than TYK2. Ellen Thomas gave a lecture that I attended some time ago, and she showed this clearly. But this may be a disadvantage also, because it seems that TYK2 inhibition has less side effects than JAK1/2 inhibition. And one of the problems that, for instance, one of the JAKs is related to hematopoiesis. So one of the complications of long-term JAK1/2 inhibition that affects JAK3 is anemia, and you don't have this with TYK2 inhibition. I think we'll need to test both separately and see which of them, if any, bring some positive results.
Yeah, that's very interesting. I wondered if we wanted to talk a little bit about the chromosome signature during the prodrome. I mean, I know you guys looked right during the infectious stage, but can you speculate on the axis of the chromatin signature during the prodrome? As we're leading up to the actual diagnosis, do you think it's just continual, or is it sort of like an on-off thing?
We're asking all the difficult questions.
Sorry.
We have the short answer and the long answer.
Okay, perfect.
We don't know. The long answer, I think we just know if it is an on and off process, as for instance in multiple sclerosis.
Yeah.
We just don't know. And I think one of the great limitations in the field is that we cannot image beta cell mass, and we cannot image inflammation in vivo. For instance, in multiple sclerosis, you can follow the process by doing MRI, and you can see when the disease is flaring. In type 1, we just don't know. But this is something I was thinking that, Jennifer, if you are doing serial biopsies, you may be able to follow, particularly if you are doing single cell RNAseq.
Yeah,
We
could add different analyses certainly to this model. It's a very rich model and reproducible, and we've already been able to gain so much data, and then see how it parallels with what we believe is going on in humans.
I guess I'd also interested in asking you guys, do you think you can use the PD-L1 blockers just during the viral course, just in a short burst for those who have genetic susceptibility and avoid the long-term use of immune sort of suppressive drugs? Could it be like, like a one and done? Or do you think it's kind of like you're going to have to monitor, this is totally hypothetical, of course, monitor someone and then dose as you go?
I think the PDL blocker is having the opposite effect. So it would sensitize the beta cells to the immune system. So this is what is observed, unfortunately, when these agents are used in patients with cancer. So some of them develop type 1, suggesting that perhaps some of the beta cells of these patients are hanging on thanks to PD-L1. So the idea wouldn't be to down-regulate, but to up-regulate PD-L1.
Sorry, that's what I meant.
But this would need to be targeted. So we would need to have a way to deliver to the beta cells, because if by any bad luck this patient has a early stage tumor, the last thing that we want is to upregulate PD-L1 systemic. So it's a delicate balance.
What do you think about this role of HLA-E? I know there's a lot of talk about HLA-G for a while, right? Because it's upregulated in the placental trophoblast, and then it was found in the beta cells, and was perhaps thought maybe it was some kind of protective, added a protective element to beta cells. But I hadn't heard that much about HLA-E, and you guys have this HLA-E was in your upregulated set.
Yeah, this was a RNA sequencing and proteomics finding, and then we collaborated with Will Morgan and Sarah Richardson. So they did histology of the pancreas of patients with type 1, and they saw an upregulation of HLA class C in beta and alpha cells, but only when there were still beta cells remaining. So there seems to be a crosstalk between the cell types. We are assuming that the function is protective, because it has been shown in other circumstances that HLA-E inhibits natural killer cells and even CD8 T cells, but this remains to be proven.
Is there anyone else in the audience who would like to ask a question?
Yeah, I had a question regarding the TYK2 inhibitor in both and also the interferon alpha receptor inhibition. So I assume it also obliterates the upregulation of PD-L1 and HLA-E. It also blocks that upregulation, right?
Yes.
And you also already sort of answered a tangential question. I was wondering if, rather than targeting the new onset type 1 diabetics, targeting the patients who have developed diabetes in response to PD-L1 blockade. But it makes sense if you do that, you could harden the tumor basically, right? You could prevent the tumor cells from dying off. But I was wondering if that would be a better risk-benefit ratio, a population where you could try it first rather than new onset diabetes. But thinking about it more, it might be counterproductive.
Jennifer, you want to answer?
You're breaking up a bit. Yeah, certainly, with administration of PD-1 inhibitors, we see, increased frequency of T1D, but other autoimmune diseases, vitiligo being apparent, and certainly in vitiligo, there are ongoing studies with JAK inhibition. So, I haven't really thought about using it as a preventive strategy, but yeah, I think being careful of untoward consequences or potentially impeding the original function of the PD-1 inhibition, it's a balance, and that's something that could probably be studied in an animal model first, and to see what the overall effects would be.
Well, it would be additive for the tumor, right? We would further downregulate PD-L1, so in that sense, it would be positive. But you could worry that if you have an immune reaction towards the tumor, that you're preventing the cell death by blocking the inflammation, maybe. I also wonder, so if you, I don't know this, if you treat NOD mice with PD-L1 blocker, what happens?
You increase very much the prevalence or the incidence of type 1. And there were some studies, this was actually 5-10 years ago, where they did the opposite. They overexpressed PD-L1, and there was a protection. So in the mouse model, it's clear that PD-L1 is protective, and deleting PD-L1 is bad news, accelerates disease.
But you talked about a mouse study, so I'm assuming that's NOD with the TYK2 inhibitor. So it seems that the balance there is towards.
Yeah, this was not tried. There was no study, to my knowledge, where PD-L1 inhibition or PD-1 inhibition was combined with TYK2 or JAK1 or even interferon receptor. This would be an interesting experiment to do, to see if this could delay the start of the disease. But to my knowledge, nobody has done it up to now.
Okay, here's a great experiment. Just waiting to be done. Thank you, Christian. That was great. I want to also bring up the fact, I know, I'm sure everyone knows that Jeff Bluestone has formed his company, Sonoma Biotherapeutics, and they're hoping, right, they've got a whole suite of Tregs and hoping to rebalance the Treg situation in type 1 diabetes. What is your thought, what might happen if Tregs, are now rebalanced? Is that going to be, how can that improve the landscape that's already been disrupted at the chromatin level?
My first thought would be, is it enough? The immunology is so complex, and we know that Tregs are an important part of the whole process, either for initiation of autoimmunity or changing that balance back. but I would, because there's just so many immune populations that are altered, and we're just, still learning about that, and, the timing, and when would be the appropriate time to introduce Tregs. I think it's definitely worth, following up and seeing what happens. But that would be the first thing that jumps to my head.
Yeah, no, that's an excellent point. The timing. Timing is everything.
It's funny, I have a collaboration with Dr. Kizi Tang, who actually was at Sugar last week.
Yes.
And one of the things that we are trying to do is to image beta cells. So we identified some surface biomarkers that are beta cell specific or nearly specific. And we are using this to try to image beta cells, but Kizi is using them to help the delivery of the T regulatory cells to the islets. Mm. So the hope is that this would make them more targeted and larger number of Tregs would home to the islets. But I share Jennifer's concern. So I think this will need to be tested and see what happens, but it may not be sufficient. One problem is that this is something that Chantal always say. I think in type 1, it will not be a one close fits all. This is a multifactorial disease, which is probably, there are a lot of individual differences. So I think we probably will need combination therapies using different drugs at different stages. And it's unlikely that all therapies will work for all patients.
Can I just ask, we're getting to the end of our time. This has been a fabulous discussion. I just have a question, if anyone, if either of you know, or anyone in the audience, is there an epigenetic signature repository for T1D patients across the, across the board, new onset, those at risk, one autoantibody, two autoantibodies, those who've had it for 10 years, whatever. You just wonder, like, as you've been talking about, once the chromatin opens and genes are turned on, what's that signature? I mean, is it transitory, or is it just like a solid signature, one and done?
From our own, that is transitory. So you see, for instance, when the human beta cells are exposed to interferon alpha, after 2 hours, you have 4,000 areas open, and after 24 hours, it went down to 1,200. I know that Klaus Kessler and colleagues, they are doing a detailed study of beta cells from type 1 diabetic patients, and one of the goals was to identify the epigenetic signature. But it has been very difficult to obtain enough beta cells. But perhaps it would be worthwhile to invite Klaus and his colleagues to one of these sessions.
Yeah, no, that's a great idea. He's attended, I believe, one of our State of the Sciences, but I would like to definitely have him join us and talk about sort of creating. I think the more mapping that can be done about the response of the beta cell and the immune signature across the disease during the prodrome, once you have it in place, when you can really get, sort of a cartography, a landscape of what's happening, that will give a lot more information to really understand sort of the snapshots that are seen. Yeah, so all right, he's next. He'll be tapped next and you'll be to blame. Thank you again for chatting with us. Are there any other questions? If not, I'll say thank you both so very much for joining us today. It was just a phenomenal discussion. You're both have done some amazing work in the field, and I can't wait to see what comes from your laboratories next. I will be looking for your next paper. Just some great work, and thank you again.
Thank you for having us. It was a pleasure to discuss with you and the other members of the audience.
Yeah, wonderful discussion, very thought-provoking, and the timing.
Thank you.
Thanks again. Have a great rest of your day, everyone.
See you.
Bye-bye.